Start. Hello and welcome to yet another Know the Nobels Forum. We had conducted the Know the Nobels Forum for all the disciplines in one go on the Nobel Day 2024, which falls on 10th of December every year, where the highest accomplishment in the fields of sciences and social sciences and literature and economics as a part of Nobel prizes are given away in Stockholm, and the Nobel Peace Prize we all know is given in Oslo. So this series of Know the Nobels Forum is about a deep dive into the Nobel Prize winning phenomena and the personalities behind it. And the entire purpose somewhere is to accentuate that noble aspiring learning in the individual classrooms, because it's a lifelong pursuit of scientists, uh, uh, where postdoctoral is something which is the basic minimum kind of a degree which we can see all through. So it's a lifelong endeavor for education, for research, and a pursuit for knowledge. And as Sir Alfred Nobel in his will talked about that all these prizes they are given to all those scientific discoveries, to all those phenomena which have brought the greatest benefit to the mankind. We'll start this series with the Nobel Prize in physiology or medicine, as they say. I'm Raghav Mittal. I'm the director programs and research at ReTHINK India Institute, and I'm joined by our president, Dr. Surbhi Vaish Mittal. So welcome, Dr. Surbhi, and Dr. Surbhi being a medico from the prestigious All India Institute of Medical Sciences would be able to guide us more and better into the innate dynamics of the human body. And I think physiology and medicine, these are the terms which are interchangeably used by the Nobel Committee. So, Dr. Surbi, just to start with, why physiology or medicine? They're used interchangeably. That would be quite trivia for our viewers to know and understand.
Well, you see, first of all, greetings on this Nobel Forum, and where we are discussing the Nobel Prize in Physiology or Medicine. Physiology, as a subject, is a very important branch of medicine. Of entire studies of medical studies, physiology talks about the functions and mechanisms, the processes, how they function, how the body works, say the circulatory system, how the heart is pumping the blood to the various organs, how breathing is happening, how the eye is capturing the image and it is coming on the retina, being relayed to the brain, how the perception is happening. So the entire processes and functions of the human body, how it operates, the biology, the physics of it, the chemistry of it, the biochemistry of it, that process mechanism is physiology. And this is of fundamental importance because something which is having physiological importance is so very profound in the understanding of the human body, of therapeutics, of pathology, which is abnormal, body structure or histology. So this is important to understand how we are, actually the functions are happening in the body. So physiology, medicine, when we talk of medicine is a broader term, so you can say physiology is a subset of medicine, and internal medicine is a subject wherein we talk about the various therapeutic aspects, how to treat the disorders. First, what are the disorders, how we treat them. So these Nobel scientists, that is why they've laid importance on the discoveries in physiology, because this really tells us about the fundamentals of the process itself, and
Mhm.
that will have profound impact on various therapeutic strategies, treatments, discoveries.
Thank you, Dr. Surbi. And this year's 2024's Nobel has been given to two very eminent laureates, which is the terminology in the Nobel parlance, to Victor Ambros and Gary Rufkun. and their collective work has been for the discovery of microRNA and its role in post-transcriptional gene regulation. So we'll come to the depth, the nuances of the Nobel Prize winning phenomena, and we would also first and the foremost try to understand the personalities behind the discovery of this Nobel phenomena for that matter. Just as a quick recap that all Nobel Prizes in physiology or medicine, so largely there are around 115 medicine prizes have been given so far to 229 medical laureates. So on an average, you can say that every Nobel Prize has gone to at least two to three. Nobel Prize is the maximum; it goes to three recipients in one go. 13 women Nobel laureates, and the youngest laureate has been at the age 31, and the oldest laureate has been at the age 87. So, the entire scientific discovery, if we just see it across, and this is on the Nobel Prize website itself, and the whole purpose of Rethink India Institute to do these Know the Nobel forums every December is to inculcate that love and that admiration for basic scientific processes and for advanced learning. And I think this love for lifelong learning can only be inculcated right in our early classrooms in our K-12 schooling, whereby we all have our fascinations कि मुझको ये बनना है, मुझको वो बनना है, तो मुझको साइंटिस्ट बनना है, मुझको एक नोबेल लॉरिएट के लेवल तक इंस्पायर करना है. I think that is something which that sanskar has to be somewhere impregnated right into our classrooms, and especially by our teachers. And that's why we also run a community called Noble Nurturing Teachers. We would be coming to that in further detail. So, any vital aspects, if you see, year on year, they have been done like 2023. Nobel Prize went to for the discovery concerning the nucleoside based modifications that enable the development of effective mRNA vaccines against COVID 19. 2022 went for the discoveries concerning the genomes of extinct hominins and human evolution. 2021, if we can see that for the discovery of receptors for temperature and touch, how human body perceives, receives the human touch and temperature, and that's what the fundamental definition of physiology, what Dr. Surbhi just enunciated, goes a long way. For the discovery of hepatitis C virus, this was 2020 Nobel Prize in Physiology or Medicine. 2019, it was for discoveries of how cells sense and adapt to oxygen availability in medical sciences, if you see, and in many of the popular uh, web series or things like if they say there's a term, technical term, I think Dr. Surbi will tell, if oxygen is not received to the brain cells, it really goes dead. So then 2018 was all about discovery of cancer therapy by inhibition of negative immune regulation. So cancer research is going a big way. So any great advance, if we have to just witness, I think nobelprize.org is the portal to which all our science faculty have to really converge and bring about that popular interest in sciences amidst the students at large. We talk about STEAM, science, technology, engineering, mathematics. Then obviously the term A has got arts, and then some of the schools are trying with stream reading. They are putting it across in sense of like literature for that matter. So Nobel is a place whereby we can really generate that popular interest amongst the young learners as to what lifelong learning is all about, and how whatever advances we are all enjoying in terms of human technology or sciences, it's all because of these lifelong endeavors. So now coming to the twin Nobel recipients of this year, we will like to reiterate that Nobel Prizes are announced in the month of October, and different Swedish institutions whom Alfred Nobel gave that mandate, they go through a elaborate nomination process which goes on for almost a year, and then they carefully select. And this is all out here. We would really urge our viewers that they should really go through this process. And 7th of October 2024, it was announced that this award goes to Victor Ambrose and Gary Rufkun. And in short, the phenomena is just the discovery of microRNA and its role in post-transcriptional gene regulation. Before we start on the whole phenomena, genetics 101, I think Dr. Surbhi will tell us more as to there are two processes, transcription and translation. I think this is a kind of a revision as well for the students, especially for the pre-medical ones, to really go through these Nobel phenomena, Nobel Prize winning phenomena. Before we embark upon this entire abstruse, interesting scientific discovery. And one more point, that these discoveries would have been done almost 30 years back, 3 decades, 4 decades back. Nobel committees, they recognize it once these discoveries have got established, stabilized. So the whole work of Victor Ambrose and Gary Rufkin was in 1993, and we are in the year 2024. So we can see that 30 years post their great work, what they did, the scientific community, it tested it took it to a different level, and then they have been given this award. So quickly, a bit about the two Nobel laureates for 2024. Victor Ambrose was born in 1953. So we can say that 70 years plus, after 70 years, that's where Nobel has come to him. He was born in Hanover, New Hampshire, USA. Most important is the educational journey, and that is something which our faculty members can really, I would say, impress upon the young, impressionable minds that this is what it takes to get a Nobel. And as Pierre de Coubertin, the founder of the modern Olympics, that's an inspiration we can take. That it is not about winning an Olympic medal. The spirit is all about aspiring and striving. And I think that is what human life is all about. Even if we are in grade 6th or 7th or 8th or 9th, 10th, 11th, 12th, I think that's where this passion, every year we can really bring it across with the Nobel laureates in that very subject. Look, this is the life which has now got globally recognized. All of a sudden, they have become the global heroes, and they are the true heroes more than any other profession for that matter, because they change the human lives in more than a singular fashion by their great meanderings into the workings of Mother Nature. So, Victor Ambrose, he received his doctorate, PhD, in Massachusetts Institute of Technology, Cambridge, MIT, the MIT, we all know about it, in 1979, where he also did postdoctoral research. Again, you see the stint of around 6 years, 79 to 85. Postdoctoral research is something, if we just plot, we will find it in the bio notes of every Nobel laureate for that matter. He then became a principal investigator at Harvard University. So, after the postdoctoral degree, the real research career started off. And in 1985, he became a principal investigator, and he was a professor at Dartmouth Medical School from 1992 to 2007. And he is currently, there is no superannuation for academics, for researchers, for scientists for that matter. ऋषियों की कोई एज नहीं होती, and he's Silverman Professor of Natural Science and Professor of Molecular Medicine at the University of Massachusetts Chan Medical School in Rochester. And one of the GK challenge we can give it to our students. Oh, if you have to really aspire for higher education abroad, just have a look at each of these institutions for that matter. Let them do a web search. Let they understand the rigor of those academic programs in these premier institutions across the globe, and come back. So, education in the classroom is all about inspiring. Delivering the lecture is one part of it all, but personalities are someone which really inspire. us to perspire for such a illustrious lifelong journey. The other Nobel laureate for 2024 in the field of physiology or medicine, Gary Rifkin, he was born in Berkeley, California. In 1952, so they are almost of the same age, we can say so. And he received his PhD from Harvard University in 1982, and he was a postdoctoral fellow, again a postdoctoral fellow at MIT between 1982 and 85. So somewhere there is a convergence uh, of these two scientists, these two Nobel laureates. Perhaps they partnered. So this is the kind of curiosity we can really build it across, that they developed their partnership here. And he became a principal investigator at Massachusetts General Hospital and Harvard Medical School in 1985, where he is now the professor of genetics. So before we
One thing
embark on,
I would like to, one
yeah,
thing
yeah.
I would like to
So,
just
yeah,
come
I
in
will
here.
just bring you, yeah. So,
Yeah.
Dr. Surbhi, please carry on. Yeah.
Yeah, I would just like to come in here when we talked about the two scientists, if you can come back to that, where
Mhm.
their educational journey was written and their professional, something which is very, very important to note here is that, teamwork and collaboration. Very often in the scientific fields, we notice that keeping knowledge to oneself, not sharing one's, observations, findings. Sometimes, you have to, one has to understand that knowledge sharing always adds to the build-up. So this is something, and if you see the Nobel laureates, they have all collaborated somewhere. So the drive is not, the goal is not that I should get the Nobel Prize. The goal is the welfare of mankind. The goal is the discovery. So
It's a lifelong passion, what
the
Dr. Surbhi has
lifelong
said
pursuit.
about. It's real
Yeah,
altruism, we can say so. More than
absolutely.
money, it is the knowledge. भारतीय विधान में हम कहते हैं महाकाली, महासरस्वती एंड महालक्ष्मी. तो महासरस्वती का जो भाव है, द नॉलेज गॉडेस ऑफ नॉलेज, इट इज शी इज आई वुड से bringing the power and the wealth together. So it's quite an intertwined precept. And just that the illustrations for this, they have been done by the Nobel Committee for Physiology or Medicine, and illustrator, we really like to acknowledge Matthias Carlin. This is the way in biology or in physiology we have to draw a lot of diagrams, we have to explain it across something we can learn. We are really passionate about biology, I would say. Even the students who really want to pursue medicine as a career, biology in their plus two level, perhaps they can really go to each of these Nobel Prize winning phenomena to really get enthused and inspired by each one of them. And the Nobel Assembly, which is consisting of 50 professors at Karolinska Institute, this Nobel Assembly, it awards the Nobel Prize in Physiology or Medicine. And its Nobel Committee, the Nobel Assembly at Karolinska Institute, Nobel Committee, it evaluates the nominations, and since 1901, the Nobel Prize has been awarded to scientists who have made the most important discoveries for the benefit of humankind. It is not for any nationality, it is not for any corporation, it is for the humankind per se. So let's come to the Nobel Prize winning phenomenon in physiology. And so this is a fundamental principle. Again, the discovery of microRNA, this is one precept, and its role in post-transcriptional gene regulation. So, Dr. Surbhi, what is transcription? Just a quick review, quick recap, so that even our a sixth grader, seventh grader, eighth grader student, and perhaps it's primarily for the faculty members, that they can take it back to their classrooms and link some of the lessons out there to the students' welfare. Please.
So, in genetics, the human genome has been mapped. We know that the human genome, we've identified some 20,000 genes already. So now the genetic process is important to understand from this point of view, that there are two processes, transcription and translation. Yeah. So DNA, or the genetic material, primarily comprising of the DNA, the ATGC sequences. What is DNA? We know deoxyribonucleic acid, and there are, genetic sequences which form the gene. So this DNA, when it is copied into the genetic coding, is copied into messenger RNA. That process is known as transcription. And then the second part of the process is translation. When this messenger RNA, which has taken the instruction from DNA or the code or the command, you can say, like a command we use when you print a document, you send a command to the printer from the computer, and that the printer then prints the documents. So the command has been given, and now the next stage, when the actual protein production is happening, is translation. When the messenger RNA is based on the code which the DNA, from the DNA, the genetic material it has got, then the protein production is happening. Now, what is protein production? How it is important? Proteins are the central building blocks of almost all the body processes, hormones, neuroreceptors, um, cardiac, receptors, antibodies, you know, biomarkers. Protein molecules are everywhere. Amino acids, we might have all heard the term amino acids.
Mhm.
So what they have discovered is this post-transcriptional gene regulation microRNA. So microRNA is a kind of a gene regulation molecule which is acting at the level of the post-transcriptional process. So
Before
one
the real production of protein is
protein,
happening, before the
yes,
translation happens. So let's come to the background. Dr. Surbi gave quickly in Genetics 101. So it honors these two scientists for the discovery of a very fundamental principle. So there is an applied thing which we see in technology and engineering, but Nobel sciences, these are very fundamental precepts, fundamental principle governing how gene activity is actually regulated, how the body commands and controls the gene. What is the Mother Nature has really given it across in that microcosm of ours, which were really so very fascinated about it. So, what they say is that the information stored within our chromosomes can be likened to an instruction manual for all cells in the body. Now, how many cells we have? I think Dr. Surbi has a quick recap. There are trillions of cells in the body for that matter. So, sabhi cells ke liye we have the information stored in the chromosomes. It's for same for everyone. Now, every cell contains the same chromosomes. And so every cell contains exactly the same set of genes and exactly the same set of instructions. Now, yet, the question what perplexes the scientists is that yet different cell types. Now, in our body, we all have studied biology 101. That different cell types are there, such as muscle cells, nerve cells, but they have very distinct characteristics. Now, why it is so? So, how do these differences arise? Now, this is the scientific curiosity. Why these differences are there when the genetic material is almost the same? So, the answer is actually it lies in gene regulation. Regulation, as we all see, there is a regulator in governance and things like that. So, body also at the genetic level, there is a regulation, traffic regulator, there are regulator of accounts, some precepts we have understood in the layman colloquial language. So this gene regulation allows each cell to select only the relevant instructions. So the body has a mechanism, but this mechanism had to be discovered. It's not an invention. It happens. It happens in the billions of people who are on the planet, who were there and who would be there. It's a natural process. So this gene regulation ensures that only the correct set of genes is active in each cell type. So different category of cells. So the gene regulation function, it makes sure that only the correct or the apt set of genes are active for each cell type. So this is the fundamental understanding which we had. So what Victor Ambrose and Gary Rufkin, they were actually interested in how different cell types develop, like when the embryonic, when a mother is really rearing that child, how this development actually happens. So they discovered microRNA. So microRNA is distinctive from the messenger RNA. I think, Dr. Surbhi, we would really like to poke you as to the difference between microRNA and the messenger RNA. Uh,
So microRNA is a non-coding RNA, first of all. It is not having a code. It is a microRNA is a code is a RNA sequence which is binding to the messenger RNA. When it binds to the messenger RNA, it blocks the certain sequence of protein production. So in that way, it is a gene regulation factor.
Hmm.
It is a catalyst, or you can say it is a factor which is
So in
resulting in gene regulation.
so in colloquial terms, we can understand that microRNA is a subset of mRNA.
Not really a subset, but then yes, it is a kind of a RNA,
Mhm.
but messenger RNA is of course having a coding sequence because that it is taking from the DNA. You know, the coding sequence is copied from the DNA to the messenger RNA. Messenger RNA has a particular code that only is translated to protein. So
Okay.
microRNA is is a kind of a tiny RNA molecule which, when it binds to the messenger RNA, it alters the protein production.
Perfect. Let's we will understand it a bit later. So they have discovered actually something called microRNA, which is a new class of tiny RNA molecules that play a crucial role in gene regulation. Now, their groundbreaking discovery revealed a completely new principle of gene regulation that turned out to be essential for multicellular organisms, including the humans. Now, it is now known that human genome codes for over 1000 microRNAs. We have 1000 odd microRNAs in the entire human genome. And their surprising discovery revealed an entirely new dimension to gene regulation. So, what is that? What is that new dimension to gene regulation? And it is a surprising discovery. This is again a very phenomenon that curious scientific research. It was not mandated. It was not a purposeful discovery like penicillin. It happened through a curious scientific process. And many of the Nobel Prize winning phenomena, if we will just go through the history of it all, they are all out of serendipity as well. You were not meant to discover it, but it happened by chance, by luck, by whatever we can call it. So microRNAs are proving to be fundamentally important for how organisms develop and function. So it's very fundamental that microRNAs are regulating the genes across different classes of cells, and that is what makes organisms develop and function. So, what is essential regulation? So, this year's 2024 Nobel Prize focuses on the discovery of a vital regulatory mechanism used in cells to control gene activity. Genetic information flows from DNA to messenger RNA. So messenger RNA and microRNA, we have to make a distinction. So genetic information flows from DNA to messenger RNA via a process called transcription. Dr. Surbi has explained it well. And then on to the cellular machinery for protein production. So they get an instruction, RNA, messenger RNA captures that instruction and it goes for to the cellular machinery. She gave a very lovely example of a printer. We gave a command from our computer and the command is taken by the messenger RNA. And printer is actually the cellular machinery and the output is some kind of a protein, whatever kind of protein we have, right?
Absolutely.
Their messenger RNAs, which are mRNAs, are translated. भाषा का ट्रांसलेशन एक बात है, पर at the cellular level we talk कि translation is actually production. Translated so that proteins are made according to the genetic instructions stored in the DNA. Somewhere or the other, why we call it as translation in the at the cellular level, कि कोई बाहर से मटेरियल नहीं देते, हम जो खाते हैं a lot of it all gets broken down into proteins, amino acids, so on and so forth. Raw material cells के अंदर है. तो वो एग्जिस्टिंग किसी मटेरियल को ट्रांसलेट कर देता है, बदल देता है एज पर द एमआरएनए इंस्ट्रक्शंस। इसीलिए संभवतः कोई बाहर से, जैसे मशीन्स, हम ह्यूमन मशीन्स देखते हैं तो इनपुट आ रहा है, मशीन है, और आउटपुट जा रहा है। पर ये सेल्यूलर लेवल की जो मशीन्स हैं उनके अंदर वो मटेरियल स्वतः विद्यमान है, तो वहां वो ट्रांसलेशन हो रहा है समवेयर और द अदर। 3D प्रिंटिंग, अभी हम जो बहुत एडवांस टेक्नोलॉजिकल लेवल पे जा रहे हैं, आई थिंक this fourth industrial revolution, the convergence, whatever is happening of all the technology, where human biology, where even the biological functions are coming at the point of convergence. These terms, see, lot of comprehension is stored in these terminologies and their contextual usages, which we have to be very, very careful and curious about it, because scientific discoveries are all about curiosity. It is something to relish, to devour. It's like your big cup of ice cream, which doesn't have to be eaten in just 5 minutes. It has to be devoured slowly and gradually, and that's where the whole taste, that whole learning, that whole love for learning develops. So since the mid 20th century, several of the most fundamental scientific discoveries have explained how these processes work. So, from DNA, messenger RNA is taking the message and instructing the cellular machinery for protein production. And friends, I tell you, even at this stage of advancement, nobody can give you a complete algorithm as to how our cellular machinery actually works. So, one small dimensions. So, Mother Nature is so very complex that the whole human great minds are really unraveling it layer by layer, peel by peel. So nobody can take that boast that we understand it fully or we can replicate it fully. We can only take inspirations of one dimension or the other and make some contribution to the humankind. So these great processes we can all go through, as I showed you at the very head start, that this is what from 2001, if we just go again, because this fundamental understanding is important. The first Nobel Prize went for the serum therapy, especially its application against diphtheria, by which Behring opened a new road in the domain of medical science and thereby placed in the hands of the physician a victorious weapon against illness and death. So the entire history of human scientific evolution on the frontiers of physiology or biology can be just mapped on this lovely webpage at nobelprize.org, that how it all progressed. I think that is again a subject matter which the history of biology, perhaps that's what the biology educators can really impregnate into their students. And as NEP talks about interdisciplinarity, so why not the history of biology, how the human biological understanding or the physiology or medicine has grown, how we have become so very advanced race on the planet. I think that is something we would like to impress upon from this forum. So our organs and tissues, they consist of many different cell types. This is again a reiteration, and all with identical genetic information which is stored in their DNA. However, these different cells express unique sets of proteins, which again is by the characteristics. So proteins encapsulate that different characteristics. So now how is this possible? How is this possible? The answer lies in the precise regulation of gene activity, so that only the correct set of genes is active in each specific cell type. So there is a natural mechanism which was discovered by these two laureates. So this enables, for example, muscle cells, intestinal cells, and different types of nerve cells to perform their specialized functions. So, like if we just come from the reproduction part of it, that how a simple zygote it formulates, I think that's biology 101. But then how the cells differentiate, पृथक पृथक प्रभेद कैसे प्राप्त करते हैं वो, how it all emanates, what is that process? I think that is where these two Nobel laureates put their brains into. It was a scientific question for a long time, but then these postdoctoral people at MIT, they really unraveled it. So, in addition, gene activity must be continually fine-tuned to adapt cellular functions to changing conditions in our bodies and environment. It's an ongoing process. So, gene activity must be continually fine-tuned. It's like docking of the spaceships, spacecraft at the molecular level, at the biological level. So, gene regulation, it goes awry, it can lead to serious diseases such as cancer, diabetes, or autoimmunity. So, this is where the application of this discovery comes into place. That a gene regulation is happening. If its fine tuning gets early, the diseases come. This is the very, very fundamental understanding of diseases. Pathogens की बात हम लोग करते हैं, but at the genetic level, ये जो chronic diseases हैं, and like cancer is now spreading like diabetes, like an epidemic in a way. We can see so by the number of cancer hospitals opening up. So something is really happening at that very fundamental cellular level. So therefore, understanding the regulation of gene activity has been an important goal for many decades. So scientific discoveries doesn't happen overnight. The eureka happens overnight, but जो उसके पीछे तपस है, the meditation, the experimentation, the modeling, it takes decades and decades. And as Dr. Surbhi said very beautifully, it's a team effort. In this case, these are two almost of the same age, but multiple generations invest their intellectual capabilities, their pursuits to come up with a eureka moment all of a sudden. And that's where the biggest benefit to mankind, that's where Nobel is so very pious and sacred. Sir Alfred Nobel developed his whole fortune by inventing dynamite, which got used more as a weapon than a peace process. But then he felt that he should use his earned endowment to institute something like that, which is being celebrated so very much out here. So just see, human genome, it contains 20,000 genes. This is a simple instruction. The flow of genetic information from DNA to mRNA to proteins. This is the whole process. The identical genetic information is stored in DNA of all cells in our body. This requires precise regulation of gene activity so that only the correct set of genes is active in each specific cell type. So Matthias Karlin has tried to illustrate it in such a elaborate fashion. So this is the human genome. It contains around 20,000 genes, and this is where a gene, the DNA is out here. So mRNA, the transcription process, the genes, you see the color coding out here. So then it does the translation. Proteins are produced and the cells get differentiated. हम लोग सभी differentiation करना चाहते हैं कि we are all unique. कहते हैं ना कि every person is unique. How? If the biology, it was just a sperm and a ovum, just at the production level, in the human aspect, how everyone grew out to be in such a spectacular, unique fashion. So again, this is another gene DNA. It mRNA carried the message, and then a different protein got created. Now just see the simplicity of this illustration. We love our NCRT books for these very illustrations. I think this is where Nobel people come into parlance की कहने को तो बहुत बड़ी बात है, but then they try to abstract it out in such a very, very fundamental fashion. So third DNA, yellow one, you see, third messenger RNA, it creates a different set of proteins and the cells take a different form. So different categories of cells. So this entire transcription and translation into differentiation of cell types is the whole process of gene regulation. So in the 1960s, it was shown that specialized proteins, which are known as transcription factors, can bind to specific regions in DNA and control the flow of genetic information by determining which mRNAs are produced. Since then, thousands of transcription factors have been identified, and for a long time, it was believed that the main principles of gene regulations had been solved. So, somewhere or the other, it was a settled problem, perhaps. That was the notion in the scientific community. However, in 1993, I think uh, 30 years back, this year's Nobel laureates published unexpected findings describing a new level of gene regulations beyond the transcription factors, which turned out to be highly significant and conserved throughout the human evolution. Now, what was that? Let's come to understanding that. So, research on small worms led to a big breakthrough. So, in the late 1980s, Victor Ambros and Gary Rufkin were postdoctoral fellows in the laboratory of Robert Horwitz. So, ये इनके गुरु थे, जहां पे ये दोनों पोस्ट डॉक्टरल फेलोज थे. एंड इनके गुरु को भी नोबेल प्राइज मिला 2002 का. लेट्स सी 2002 का नोबेल प्राइज क्या मिला इनको. वी हैव आवर इंडेक्स आउट हियर. So they got the Nobel Prize, Horwitz, you can see that here, for their discoveries concerning genetic regulation of organ development and programmed cell death. तो ये एक अलग विषय वस्तु है. So alongside Sidney Brenner and John Sulston, we'll not go into that, but they were the students of a Nobel laureate in 1980s. So in Horvitz's laboratory, they studied a relatively unassuming 1 mm long round worm, and what's name is C. elegans. This is a round worm, C. elegans, which is just 1 mm. So despite its small size, C. elegans possesses many specialized cell types as nerve and muscle cells also found in larger, more complex animals. So from the animal kingdom, they extrapolated it across to the human behavior. So this 1 mm chota sa C. elegans, it possessed specialized cell types, nerve, muscle cells, and making it a useful model for investigating how tissues develop and mature in multicellular organisms, because a lot of experimentations just cannot be done on human bodies right away. We have an entire compact of experimentation, and then we evolve it across. That's how the especially the research in the biological space happens. So Ambrose and Rufkan were interested in genes that control the timing of activation of different genetic programs. Now, gene code is like a program. There is a legal code. There is a code in computers. We are also very fatuated about it, but the fundamental code is out there in the human body, the genetic code. That is why the word genesis, it's at the core of it all. So, ensuring that various cell types develop at the right time. So there is a temporality also out there. कि अगर मसल सेल बनना है, नर्व सेल बनना है, तो टाइम फैक्टर इज ऑल्सो आउट देयर. देयर इज समथिंग आउट देयर इन द जीन्स. So they studied two mutant strains of worms, Lin4 and Lin14. So this is a standard operating protocol in the biological research, that they create different, because scientific observation has to be done, because we cannot just go at that very cellular level. It has to be observed indirectly. That's the whole scientific process is all about. So they created mutants so that they can compare the outcomes somewhere or the other. We will come to it. So for the sake of memory, Lin4 and Lin14 are the two mutants where the genetic mutation has been orchestrated in some way or the other. That's not the subject matter. And C. elegans के दो जो mutants हैं, Lin4 and Lin14. So they studied these two mutants, and that displayed defects in the timing of activation of genetic programs during development. तो फर्क आ गया कि that displayed defects in the timing of activation of genetic programs during development. So the laureates wanted to identify the mutated genes and understand their functions. तो म्यूटेशन उन्होंने किया, उन्होंने कहा कि म्यूटेशन जो हुआ उससे कुछ डिफेक्ट आया. So Ambrose had previously shown that the Lin14 gene appeared to be a negative regulator of the Lin14 gene.
Lin4 gene, yeah.
Lin
Lin4 gene appeared to be
Lin14.
negative
Okay,
regulator
both
of.
Lin4 and Lin14, they have a complementary role somewhere or the other at the cellular level. तो यहाँ पे इनकी पार्टनरशिप भी आप देखते हो कि एम्ब्रोस ने कुछ काम किया और रुफकिन ने उसको अलग से देखा. वी विल जस्ट कम टू दैट. However, how the Lin14 activity was blocked was unknown. And Ambrose and Rufkin were intrigued by these mutants. Mutant का शब्द बहुत आसान हो गया है. We see a lot of Wolverine. There are human mutants doing a lot of functions, but at a very, very down to earth scientific level, कि सी एलिगेंस के दो म्यूटेंट्स में उन्होंने कुछ काम किया एंड दे फाउंड दैट लिन 14 उसको ऑब्स्ट्रक्ट कर रहा है. देयर इज समथिंग डिफरेंट. So Ambrose and Rifkin were intrigued by these mutants and their potential relationship and set out to resolve these mysteries. तो यहाँ पे उनका जो साइंटिफिक प्रोसेस है, द क्वेश्चन इट इवॉल्व, अब वो वहीं रुक गए. Let's solve this mystery. Now let's understand further. See, scientific understanding has to go iterative. We just read it, we will see this, then we will try to understand this, and then we will make sense of it. That's what science is all about. So C elegans का स्ट्रक्चर इन लोगों ने दिखाया है, जिसका एडल्ट साइज वी जस्ट रेड इट इज अराउंड 1 mm. ये वॉर्म है. दिस इज जस्ट अ मोमेंट. राउंड वॉर्म इज अ कैटेगरी ऑफ. तो इनका एक नर्वस सिस्टम भी है. इट हैज अ नर्वस सिस्टम का सेल है, गट है और मसल है. इतने छोटे वॉर्म के पास भी 3 प्रकार के सेल्स हैं. फिगर बी, लिन 4 एंड लिन 14 म्यूटेंट्स. तो नॉर्मल, ये नॉर्मल सी एलिगेंस दिखाया गया है. ये लिन 4 म्यूटेंट है और लिन 14 म्यूटेंट है. तो एलिगेंस का जो जीनोम है, इट हैज अराउंड
1 billion, I think.
यस. अगर हम इंडियन नोटेशन में देखें तो 1010 crores, 1 billion base pairs, ACTG base pairs. So Lin 14 is depicted in blue and Lin 4 is depicted in this reddish color. So Victor Ambrose and Gary Rufkin. So Lin 4 has so what they are trying to explain that C elegans is a useful model organism for understanding how different cell types develop. Ambrose and Rufkin studied the Lin 4 and Lin 14 mutants. Ambrose had shown that Lin 4 appeared to be a negative regulator of Lin 14. It's a negative regulator of Lin 14. This is something we have to understand. What is a negative regulator? We will key what function Lin 4 does. Lin 14 does the opposite, perhaps. So Ambrose discovered that Lin 4 gene encoded a tiny RNA, which they termed as microRNA, that did not code for a protein. Rufkin cloned the Lin 14 gene, and the two scientists realized that the Lin 4 microRNA sequence matched a complementary sequence in the Lin 14 mRNA. So this is 22NT microRNA of Lin 4 and Lin 14. So Lin 14 mRNA, they are showing a sequence CUGAC, and this is a kind of a negative. This is Lin 4 microRNA and this is Lin 14 microRNA. Let's try to understand. So this is the sequence they discovered, they unraveled that one of them is a negative regulator of the other.
So
So
basically, when Lin4 is The microRNA, which is being, coded from Lin4, you can see that it is a yellow colored microRNA, which this, when it is binding to the Lin14 mRNA, this yellow round here,
Mhm.
circle here, some part of the protein production is being blocked. So resulting protein which is being produced is altered. That's what is gene regulation,
Mhm.
negative gene regulation.
So after his postdoctoral research, Victor Ambrose analyzed the Lin4 mutant in his newly established laboratory. कहानी यहाँ पे इंटरेस्टिंग है. पोस्ट डॉक्टरल रिसर्च में रॉबर्ट हॉर्विट्स में यहाँ तक तो ये लोग आए कि ये जो म्यूटेंट हैं ये एक दूसरे के नेगेटिव रेगुलेटर्स हैं. Lin4 appear to be a negative regulator of Lin14. यानी कि उसको कोई बाधित कर रहा है. इट्स ब्लॉकिंग इट. लाइक
So,
इन मेडिकल साइंसेज देयर इज समथिंग कॉल्ड ब्लॉकिंग. डॉक्टर साहब ये ब्लॉकिंग का कॉन्सेप्ट थोड़ा सा समझाइए. बीटा ब्लॉकर्स और समथिंग ब्लॉकर्स जो आप लोग प्रिस्क्राइब करते हैं सो दैट वी कैन मेक सेंस ऑफ इट एट द सेलुलर लेवल.
so blocking, is a very important concept in biological processes. Say, for example, if you see, there are neuroreceptors. Neuroreceptors are certain protein molecules on the neurons, which would perceive the neural signal.
Okay. तो
That's
जैसे
how.
पेन किलर खाते हैं फॉर अ सिंपल लैंग्वेज. सो व्हेन वी ईट अ पेन किलर, इसीलिए डॉक्टर्स कहते हैं कि ज्यादा पेन किलर मत खाओ. तो दोस पेन किलर समवेयर ट्राई टू सप्रेस.
Yes. So they alter that receptor mechanism. That's how the pain perception is blocked. That's a kind of a blocking fundamental. Also, you can say whether we have the antigen antibody response. Again, the antibodies are blocking the microorganism invasion. What is antibody? Antibody is the body's immune response to fight against the invasive microorganism activity. So
I
again,
think
this is kind of a blocking.
this will further get clearer.
Yeah.
And as a part of the Nobel content, they also produce elaborate scientific document also, which is the scientific background, further scientific background, which further details out the whole mechanism out there. सो दिस इज बियॉन्ड द स्कोप ऑफ इट ऑल. वी हम लोग तो कहानी भी बता रहे हैं फिनोमिना कैसे डिस्कवर हुआ. सो जब हार्वर्ड यूनिवर्सिटी में आफ्टर हिज पोस्ट डॉक्टरल रिसर्च ही वेंट, सो methodically mapping means ये sequence, this is the sequence mapping which is done by the genetic scientists across. So methodical mapping allowed the cloning of the gene and led to an unexpected finding. Victor Ambrose, the Lin4 gene produced an unusually short RNA molecule that lacked a code for protein production. It was just a blocker. It did not have a code for protein production. This is a difference. So these surprising results suggested that this small RNA from Lin4 was responsible for inhibiting Lin14. Now, how might this work? How might this work? So, concurrently, Gary Rufkin investigated the regulation of the Lin14 gene in his newly established laboratory at the Massachusetts General Hospital and Harvard Medical School. So, having well-stuffed laboratories, the newly established ones, घर में जब कोई नई चीज आती है तो we all get enchanted to use it. So these two scientists, they were lucky enough to get newly established laboratories, each one of them. And unlike how gene regulation was then known to function, Rufkun showed that it is not the production of mRNA from Lin14 that is inhibited by Lin4. It is not the production of mRNA from Lin14 that is inhibited by Lin4. The regulation appeared to occur at a later stage in the process of gene expression through the shutdown of protein production. So something more subtle was happening. This is what he discovered. It was not like it the lacking of a code for protein production was done by Victor Ambrose, but his research said that it is happening at a, that's why the Nobel Prize comes at a post-transcriptional gene regulation. There is something subtle which is happening. We'll understand this. So the regulation appeared to occur at a later stage in the process of gene regulation through the shutdown of protein production. So something was blocking it. Something was uh, something was instructing that. So experiments also revealed a segment in Lin14 mRNA that was necessary for its inhibition by Lin4. And the two laureates compared their findings because they had a MIT background and they worked at the same Nobel laureate guru, and which resulted in a breakthrough discovery. Now again, जैसे चाबियां होती हैं, you have two codes, so two friends have to come together to make a breakthrough discovery. अगर अकेले हमारे रुफकुन साहब रह जाते या हमारे एम्ब्रोस साहब रह जाते तो शायद ये जो डिस्कवरी है, आई थिंक डॉक्टर सुरभि ने बहुत पहले ही इस गूढ़ ज्ञान को रख दिया था. If they would not have exchanged their notes, I think the breakthrough discovery would not happen. So the short Lin4 sequence matched complementary sequences in the critical segment of Lin14 mRNA. बहुत कुछ हमारे ऊपर से जा रहा है, बट कोलैबोरेशन की बात हमने समझी है, and there was something which was happening. And Ambrose and Rufkin performed further experiments showing that Lin4 microRNA turns off Lin4 14 by binding to the complementary sequences in its mRNA, blocking the production of Lin14 protein. So here a new principle of gene regulation mediated by a previously unknown type of RNA, microRNA, had been discovered. So messenger RNA to message लेकर जा रहा है, but there is a parallel, there is a microRNA which is existing, which is regulating it across. This is very important to know. And these results, they were published in 1993 in two articles in the journal Cell. So this is where scientific process comes. You publish your discovery, it is vetted, then it is applied upon through some technological mechanisms or things like that. So the published results were initially met with almost deafening silence from the scientific community. Although the results were interesting, the unusual mechanism of gene regulation was considered a peculiarity of C. elegans, likely irrelevant to humans and other more complex animals. तो साइंटिस्ट में एक स्केप्टिसिज्म होता है, अरे भाई सी एलिगेंस में हो रहा होगा, उसका ह्यूमन से क्या वास्ता. So that perception changed in 2000 when Rufkun's research group published their discovery of another microRNA encoded by the LET7 gene. And unlike Lin4, the LET7 gene was highly conserved and present throughout the animal kingdom. So the same phenomena was ratified 7 years after the first Lin4 Lin14 saga. And the article sparked great interest, and over the following years hundreds of different micro RNAs were identified. वो कहते हैं ना, साइंटिफिक ब्रेक थ्रू एक हो जाए तो फिर उस रास्ते पे चल के धर 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 एक कोविड की वैक्सीन आई, फिर उसके बाद बहुत सारी और वैक्सीन आनी शुरू हो गई. तो बहुत सारे हंड्रेड्स ऑफ डिफरेंट माइक्रो आरएनएज वर आइडेंटिफाइड. जैसे कि हम एटॉमिक स्ट्रक्चर में पढ़ते हैं कि भाई न्यूट्रिनोज नया पार्टिकल डिस्कवर किया, तो एट द सेल्यूलर लेवल ये माइक्रो आरएनए एक नई एंटिटी के रूप में उद्भव हुए. प्रोसेस को समझने में थोड़ा शायद हमारा इनरोड, बिकॉज दिस इज ऑल एक्सपेरिमेंटल जेनेटिक्स, वी हैव टू हैव फर्दर inroads into that. But at the level of our the purpose, our objective, इतना समझना भर काफी है. So today we know that there are more than 1,000 genes for different microRNAs in humans, and that gene regulation by microRNA is universal among multicellular organisms. So अगर हम ये फर्दर इलस्ट्रेशन को देखें, दिस इज गैरी रुफकिन्स वर्क, सी एलिगेंस लिन फोर लेट 7, इन्होंने इससे प्रूव किया ह्यूमन के अंदर लेट 7 है, और 500 मिलियन ईयर्स से ज्यादा होमोसैपियंस। सो ये माइक्रो आरएनए ही है कि कहीं ना कहीं कि मनुष्य और प्रोरीफेरा, सिरेंडेरिया, ये जो पूरा एनिमल किंगडम है, इट परमिएट्स ऑल अक्रॉस, एंड दिस इज वेयर स्पीशीज का जो डिफरेंशिएशन है, सेल्यूलर लेवल से ही तो स्पीशीज का डिफरेंशिएशन हुआ। आई थिंक फर्दर इनरोड्स विल टेल कि कैसे ये स्पीशीज का उद्भव है। तो रुफकिन क्लोन्ड लेट 7, a second gene encoding a microRNA, and the gene is conserved in evolution, ये जीन एवोल्यूशन में कंजर्व रही, and it is known that microRNA regulation is universal amongst multicellular organisms. So in addition to the mapping of new microRNAs, experiments by several research groups, so ये microRNAs जैसे कि बार बार हमने कहा, they don't have the code for production, they inhibit, they are like controllers, they don't instruct कि आपको ये प्रोटीन बनाना है, but they help the expression of a gene कि आप nerve cell बनोगे, आप muscle cell बनोगे, that is where their specialty comes into play. And the binding of microRNA leads to inhibition of protein synthesis or to the mRNA डिग्रेडेशन। तो ये एक फंक्शनैलिटी है। हम लोग इसको ऐसे जेनेटिक रेगुलेशंस को हम समझते हैं। आई थिंक बायोलॉजी फैकल्टी विल बी एबल टू एक्सप्लेन इट फर्दर, बट ये जो प्रोसेस है, ये जो छोटी सी डिस्कवरी हुई, दो साइंटिस्ट थे, दोनों ने उसी फिनोमिना के दो दो आयामों की ऑब्जर्वेशंस की, एंड व्हेन दे एक्सचेंज नोट्स विद ईच अदर, they came at a wonderful discovery out there. So intriguingly, a single microRNA can regulate the expression of many different genes. And conversely, a single gene can be regulated by multiple microRNAs, thereby coordinating and fine-tuning entire network of genes. So this is getting so very exciting. We talk about a lot of networks of computers, routers, so on and so forth. But the human body, which is one of the finest, majestic, most evolved machines, we can say, so biological machine, understanding and unraveling it's so very astonishing. So cellular machinery for producing functional microRNAs is also employed to produce other small RNA molecules in both plants and animals. For example, as a means of protecting plants against virus infections. So Andrew Z. Fire and Craig C. Mello, who were awarded the Nobel Prize in 2006, they described RNA interference, where specific mRNA molecules are inactivated by adding double-stranded RNA to cells. So this is again a very complex phenomena. But then gene regulation, microRNA is a new entity. This is something which we are making sense of it quite well. एंड टाइनी आरएनएज विथ प्रोफाउंड फिजियोलॉजिकल इंपॉर्टेंस, ये जो माइक्रो आरएनए आ गया अभी नया, दिस इज द न्यू बॉय इन द क्लब ऑफ डीएनए, एमआरएनए, एंड इट हैज अ प्रोफाउंड साइकोलॉजिकल इंपॉर्टेंस। बहुत अच्छे से मैटियस कार्लन ने इसको कलरफुल प्रस्तुत किया है। एंड जीन रेगुलेशन बाय माइक्रो आरएनए, फर्स्ट रिवील्ड बाय एम्ब्रोज एंड रुफकुन, हैज बीन एट वर्क फॉर hundreds of millions of years, the entire evolutionary journey. This mechanism has enabled the evolution of increasingly complex organisms, the differentiation of entities. And we know from genetic research that cells and tissues do not develop normally without microRNA. So abnormal regulation by microRNA can contribute to cancer, and mutations in genes coding for microRNAs have been found in humans, causing conditions such as congenital hearing loss, eye and skeletal disorders, so and so forth. So microRNA अगर tamper हो गया किसी कारण से, that is what now scientists are studying. So the expression, the gene expression would get inhibited, and that's where the medical application of this fundamental scientific research in physiology or medicine comes into play. So mutation in one of the proteins required for microRNA production result in the Dicer 1 syndrome, a rare but severe syndrome linked to cancer in various organs and tissues. So Ambrose and Rufkin's seminal discovery in the small worm C. elegans was unexpected and revealed a new dimension to gene regulation, essential for all complex life forms. So 20,000 odd genes, mRNA, and it is not producing a protein because a microRNA is out there. It has inhibited the production of that particular protein, and that's how microRNA. So it leads to development of human body at the physiological level and treating and curing cancer. I think Dr. Surbhi, ये जो Dicer 1 syndrome है, would you like to just from the applications perspective ki what kind of uh, as a practicing medico and someone who has so very deep inroads into genetics, what kind of implications you are finding going forward?
So definitely, we know that in the pathogenesis, etiopathogenesis of diseases, genetic factors has also a role to play, because many of the basis of diseases lies in the abnormal genetic functioning. It may be due to acquired mutations or certain genes which are recessive and become dominant in the fetus. When a recessive or a recessive gets, a recessive parent marries a recessive parent, the recessive becomes dominant.
So
That's
now
about
we
the
have got
genetic part.
that vehicle. The discovery of these microRNAs has
Yes.
empowered the scientific community कि भैया ये जो हजारों microRNAs हैं, ये हैं या तो देवता या दानव. मतलब they are the culprits, they are the agents of making it all happen.
So the Dicer 1 syndrome is basically a genetic, predisposition, wherein the person with that genetic code has is vulnerable to acquiring tumors of so many organs. So definitely the application part of this is there, that this microRNA production is resulting in the Dicer 1 syndrome. Cancer is one very, we should say, great challenge to the medical fraternity today, because we say cancer is curable if detected early. But then, if we go in the genesis, why the cancer is actually occurring? So, this microRNA production, of course, some of it is coming from the genetic code. And then mutations can also happen due to, environmental factors, diet, and, well, a lot of research is still going on, how stress and
So at the macrocosm level, we are witnessing, we are understanding. So human endeavor is an ongoing one. The
true.
average human lifespan is under 100. We can so these discoveries, this collective pursuit of the human scientific mind is going on and on. And I think we are now going at the like gene editing has been made possible. Messenger RNA based vaccines created. We have seen their applications. Also, the adverse effects which happened because of the mRNA carrier vaccines for the COVID-19. So I think human wisdom, human knowledge, human scientific prowess is really moving further and forward. And the Nobels are the greatest tribute, the greatest benefit to the humankind. And we at Rethink India Institute very, very fervently and very earnestly feel that these very scientific discoveries, these very Nobel Prize winning phenomena and personalities, they go hand in hand. And they have to be made a very vital mechanism, a very vital instrument for advancing the love of learning, love of advanced learning in each of these subject matters. And that's where the Nobel nurturing teachers come into play, who attune their students to the aspirational academic footprints and creative characteristics of the Nobel laureates. That is something which is so very important. This was the Know the Nobels Forum for the Nobel Prize in Physiology or Medicine 2024, which was conferred upon Victor Ambrose and Gary Rufkin. Quite inspirational stories in partnerships They studied together, then their independent researches, and how a small, tiny roundworm came to rescue the future of humankind. These are all very inspirational stories, and applied genetics as to how the mutation is happening, how the gene codes are getting suppressed. These are very technical issues. I think the genetic classes can get further pepped up by invoking these things, and we saw that how the various years of Nobel Prize winning researches are quite intertwined, quite interlinked. एक अंतर गुम्फित है ये कॉस्मोस, यह क्रिएशन, एंड इस क्रिएशन को जानने की जो क्यूरियोसिटी है, दिस इज व्हाट साइंस इज ऑल अबाउट. हमें आशा है कि यू वुड हैव लव्ड दिस सेशन. And thank you, Dr. Surbhi, for your expert commentaries and comments as a practicing medico, because medicine is an applied part of it. And these Nobel Prizes, they have been awarded because the implication of microRNA in fighting the human menace, the menace, the disease burden, the chronic diseases especially, which can perhaps be rectified for a better, for the benefit of humankind. So thank you so much. We will be back again tomorrow with the next Nobel Forums for the Nobel Prize in Physics. Do watch and do use this material to attune your students, to inspire them, to aspire them. And also, teachers are, after all, they were all teachers. They are all teachers and researchers. So, at the school level, we have to be somewhere inculcating that lifelong learning. We can create our own textbooks, we can create our own material. Whenever we are talking about 4 or 5 Nobel Prize winning phenomena, can always be incorporated. And thanks to the Nobel Committee, their generous and their stellar efforts in very simple language, in a very beautiful illustrations, they can always be copied to your smart boards and things like that. And you can really take your classroom lecture a Nobel, incorporating the Nobel narrative in some of the classrooms would be really lovely going further and forward. With this, we close today's Know the